And I wish I was as good as gardening as Lexi is. So my garden is full of hybrids. It's a real gene pool party.
Can we get, like, sad trombone noises intermixed here? Wow, wow.
Welcome to Absolute Genius, a podcast series from Thermo Fisher Scientific. I'm Jordan Ruggieri, and we are excited to kick off our fourth season of the Absolute Genius series. In case you missed our teaser for the season, I want to clue you in to some changes. We'll continue to dive into really cool science in an approachable and entertaining manner. The puns are wonderful this season, I promise you. This season, we'll be widening our lens to look beyond just digital PCR, and we'll be covering qPCR and other complementary technologies as well, along with how they work together to accelerate a wide variety of research. Another change for season 4, I'm excited to introduce my new co-host, Lisa Crawford.
Hello, listeners. I'm Lisa Crawford. If you want to know more about me, I encourage you to go back and listen to the teaser for the season, where I talk a little bit about myself. But today, we are excited to kick off our fourth season of this series by going green with a certified organic genius, Lexi Heger. Lexi is a PhD candidate in plant pathology and molecular plant sciences at Michigan State University. She's a lifelong plant lover, a budding qPCR and dPCR aficionado, a seasoned plant punster, and an all-around fascinating person to talk to. We hope you enjoy today's episode.
Well, thank you so much for being on Absolute Genius. We are so excited to have you on the podcast today.
Thank you. I'm excited to be here. It's fun to bring ag to this kind of science, I think.
Could you introduce yourself and maybe just give a little bit of background into your science and research?
I am a fifth year PhD student at Michigan State University right now. I have a dual degree in plant pathology and molecular plant sciences, or I will have one. My background is originally in horticulture, where I went to the Ohio State University. I really just loved plants growing up. I didn't really know that you could study plants as a field when I was especially in high school, right? My parents owned a plumbing company, and so I was kind of the weird little black sheep, just figuring things out, growing crazy things in the yard. My dad just they kind of let me have free will and just do things. And then took AP Bio in high school, and my teacher, who's still a mentor to me today, she encouraged me to pursue plant science, and then it kind of just took off from there. Now looking at my research, our lab is a small fruit and hop pathology lab with Dr. Tim Miles here. We focus on a lot of different fruits and then fruit pathogens, so grapes predominantly, then hops, blueberries, raspberries, strawberries. So yeah, we got it all. You just had wine, now we also have beer. There's a lot going on.
This is going to be an exciting episode. So tell me a little bit about this world. I mean, is it mostly studying plants, or are you actually studying, bacteria, virus, yeast, fungus and interactions on the plant themselves?
I try to do a mix of both in my research because I like the plant side and I also like the pathogen side. I also work with an oomycete, so that's a fungal-like pathogen. So people often forget that one. I'll call it fungal-like just to keep it, chill for people. But it's also an obligate, so right, it requires a living host to survive, right, and to reproduce. And so that actually adds a whole nother level when you're working with it in the lab. I have to keep plants alive. So when you ask that question for me as a pathologist and as working with an obligate, I'm like, yeah, I got to do both, but I always have to do both because if I want to work on this pathogen, I need to keep a plant alive.
With the worst green thumb on the planet, I can only imagine.
We kill a lot of plants. Like as pathologists, our goal is to kill plants and to save them. Like we kill them to save them.
What is your PhD centered on?
So something that was important to me was getting a PhD that was honestly kind of diverse within my field so that I could have multiple skills. So really it starts out focused on downy mildew of grape, which is the obligate oomycete pathogen I was just talking about. It is a very destructive disease of grapevine. I first designed a diagnostic tool using qPCR to differentiate these three cryptic species of Plasmaphyra reticula, this pathogen. And so we wanted to differentiate them using these molecular tools so that we could identify them in the field and do further research, right? So you got to start somewhere to be able to do those long-term studies. And honestly, in the last, I guess, even 5 years of my degree, a lot of work's popped up doing that. And so it's been kind of cool to see it develop. Then the second chapter took another turn. I do a lot of spore trapping too. So it's capturing or sampling the air to kind of identify what is present in terms of pathology, outlook. You're usually defining what pathogens are there, how much of them are there, when are they there. Eventually, people look at microbiomes. Are they interacting? You can go forever and ever, right? And so I redesigned qPCR assays that had been previously published, and I combined them into a multiplex so that we could detect these three really important pathogens of grapevine again, which was downy mildew, same one before, the Tritis cinerea, which is fungal, and then another obligate fungal pathogen called air siphon indicator or powdery mildew. Being able to capture these and then detect them is really important for fungicide programs. And then my last chapter is just looking at, well, not just, I guess, it's doing a GWAS study, so genome-wide association study, looking at a population that's from California that had known botrytis and powdery resistance, and trying to see if also there was downy mildew resistance and how that kind of relates, looking at it in Michigan environment versus California.
Are these pathogens, are they commonly tested in vineyards, or is it more just they see something is wrong with the plant, and it's just more kind of a, hey, they do we tear them up? Like what exactly happens on a practical level for the farmers?
It depends where you are. Again, it depends, always comes up. In Michigan, we have more humid environment, right, compared to California or Oregon. The testing isn't as prevalent as we'd like it to be. Usually, you would want this done early in the season. I want it like before you're really getting a lot of plant growth. Then you can say, hey, we have this here. We should spray fungicides preventatively because once infection takes over, you're at a loss at times. There's also phenological stages, right? Different development stages of the grapevine, and there's different important time points for spraying. And so it's really about using these tools in conjunction with that knowledge of like the phenological stages and knowing, okay, if we test this time, we can learn how much is here in terms of what kind of spray program can we use. And people are even, or there's been a lot of assays, qPCR assays designed looking at fungicide resistance detection. So those rare alleles, and we can detect for it's like FRAC11, which is a fungicide group. We can detect for that fungicide resistance, and then you can tell the growers, say, hey, you should steer clear of these because you're going to encourage development of resistance, which means losing control of the disease, and that's just bad. Testing is, again, very limited. There was a big grant that our lab group was in collaboration with a couple others that did nationwide testing for a few years, and it was a huge success because they were able to track resistance populations around the country, and that's huge. But when funding goes away, right, they aren't able to test anymore because the need is just it's there, but it's not great enough.
Is that what you think the biggest barrier is? Is the funding and cost, or is it education because people don't necessarily realize the importance of it, or is it kind of a mix of everything?
I would lean towards a mix of everything because when you say people, my immediate thought is what the growers know, right? But people is not just the growers, right? And so you need to advocate for the growers to the broader population. And so, but yes, I think when I think of sampling and testing, for me, the air sampling thing comes hand in hand again. And so setting up tools like that that aren't meant for or that are meant for research, not necessarily for growers, that's a whole nother barrier is like the resources is more than money, right? It's time, it's effort, it's knowledge. And so it's kind of wild. That's been a big part of the background of my PhD is thinking about what are the barriers to improvement. And it's a lot of times is that transition from research to applicability.
What I can imagine too, even talking a little bit about the workflow, you're looking at air samples, but you're probably also looking at plant and soil, or at least having particulates of those in the sample, which can pose inhibitory risks, right? Too on the workflow and on the detection end. Is that something you run across regularly as well? Any of the polyphenolics or the cell debris, the cell wall debris that can inhibit detection?
That was a big concern and thing we think about sometimes is we think about it too late. I think with plant pathology at time, like sometimes early in my projects, as a first year PhD, it was like you forget that's another part. You're so focused on the detection of just straight up detecting. But yes, that's been a large focus, and you're right. With our field, especially even the air samples, I had bugs stuck on my sampling rods. I had dirt. I had everything, and I personally don't know if it was inhibitory because I had other issues. So I had some other extraction issues, but usually the results are clean enough and they're consistent enough where we're not really seeing that issue. But I've seen other papers where they were doing the same thing and looking to see what was the inhibition. They had internal controls. I couldn't use one because I already had three probes built in. So it was just like, I guess this is our trade off, right? But that is a huge concern in plant pathology, especially people with soil. And a lot of times they focus on making sure that's not being a problem, but I think it depends on the method and depends on the extraction too. We've found, right? It's not just point blank. Do you have inhibitors?
I mean, I this coming from the lab, working with some of these different samples as well. I mean, it is a headache sometimes. Taking a step back even as well, you mentioned that you have different clades that you're looking at. Do they have different impacts on the great plants and on growing, or is there an actual like practical or like real implication of having a different clade that is on your plant?
That's the big question, and it's an ongoing question. And like I said before, there's been more publications discussing, the implications because we don't really know yet. One of the interesting things is that I talk about three clades. There are a few more, but they're not as economically important, meaning they don't infect cultivated grapevines. In the United States, we have all three of these economically important clades, but in other countries, we do not. And so there's also that. Why hasn't it moved? What does that mean? Why is this happening? The biggest clade is called clade estivalis. Biggest clade, as in the most, it's worldwide. It is very destructive, and that's the one we've always talked about. Historically, whenever we've seen literature about P. verticilla, Plasmodium verticilla, it's been about that. We just didn't know it because they've done studies looking to trace and see which clade was it that has been everywhere. Um, but we still don't really know. We tried to look at in my lab with one of my lab mates, we looked at she was doing fungicide resistance, and so we're trying to see can we associate because in cucumber and a similar pathogen, very closely related, they have seen that there is fungicide resistance based on clade, and so we were like, oh, is that something that's happening here? And as of now, we can't say that there is. Kind of surprising. I mean, from a pathology nerd side, was kind of like, dang it, thought we were really onto something, But also, those clades are different. I mean, that system is different. Cucumber is different. There's a lot of things, right? So there are more aggressive within the three clades. There's one that infects the wild grape, and that one is not, it's much more host specific. Wild grape, for one, is its main host, and so it doesn't as readily infect other cultivars of grape. So, everyone hears about like Chardonnay, which is a big vinifera, um, which is what people drink that, right? Of course, everyone knows that one. And that one can't be infected by the wild grape because it's a pure vinifera. And so, right there in my mind, it's like, okay, so maybe that clade is not as impactful. How important is it? And I don't know. It's interesting. I think this is going to be, if people have the funding for it, it'll be a lot of future studies. I got funded for a NIFA pre-doc fellowship to actually look at the effector profile of these three clades. And so that's in the works right now. We did the infection on one single host, and we're looking to see how do they compare. And it'll be interesting to see between, the one we know is pretty aggressive versus the very host specific versus one that's kind of an intermediate, but shows to be more aggressive, but doesn't exist in other places. So I think it'll be pretty exciting if it works out. Hopefully science works out for us here and get to see because in other places, they don't have all three of these, and so they haven't been able to do as much research because they don't have access to the isolates.
That's really interesting. It's cool to also hear the I don't knows, right? I think that's a cool part of science as well, right? And just hey, this is something we think there might be something here, but we're looking, and obviously there's some like you said with the wild grapes, like there's some differences, right? It just depends on the grapes that are growing or what you're utilizing. Can we talk a little bit about your publication that was just recently put out on APS? Can you talk a little bit about what you were looking at there and maybe some of the results?
So this paper was going back into the three cryptic species or clades. I'll stick with one word of cryptic species for now of P. viticula. We wanted to see again baseline. Can we differentiate them using a tool, and then go from there? During actually the process of this, another study published using a different loci. So we used a mitochondrial loci in this study. Mitochondrial usually helps us be more specific, whereas ITS or ribosomal, which is the other loci, it can be a little less. We might have increased sensitivity, but we might lose some specificity there, which is really, I know with qPCR, that's a huge trade off, right? And so something that's really important to think about. In my paper, we focused on also making this a multiplex, and trying to eliminate maybe having multiple primers, maybe just having one primer set and then the three probes. So we wanted it to be simple. It never is, but we wanted it to be simple and to be able to clearly differentiate these. The focus, in the beginning was really okay. Can we also make this quantitative in terms of like, can we accurately determine how many spores or something like in that same vein? There were some difficulties when we got into this, when we got into the mitochondrial loci. There was just some development problems in terms of, trying to make these all three work together. It was a low GC content, and so then we had some problems. It still did effectively work. We found that two of our probes were, sensitive to a level that in pathology we deem appropriate. The goal of this was really to track things throughout the season, and so we kinda did a preliminary study using data from another chapter of mine with the air sampling. We didn't realize we had multiple clades, actually, so it's really cool. In my vineyard site, we didn't realize we had 2 of the clades. We thought because every time I sampled, we only got Clade estivalis, but we actually had Clade estivalis and Riparia because Riparia isn't as aggressive, right? And so it wasn't colonizing the same way. So it was really cool to use this very specific tool and be like, "Oh, wait, we can actually detect and see the fluctuations throughout the season," which I think was, amidst the troubleshooting, that was actually really exciting to see that this can work. And although there are limitations, we can still get a general idea of how much is present. And then we wanted to enter the world of dPCR. I was the first one, so I got thrown into the fire, but it was a cool fire. I learned a lot through this. When I went to write my discussion, I was like, okay, maybe this my assay design wasn't perfect for dPCR because of these problems I had prior to it in terms of troubleshooting and the lower efficiency because of the loci and such. But you can really see the utility of it. With I was looking at again kind of the rare alleles a little bit, or just very like specific SNPs, and so I was able to see those changes, and I think between 2 of the assays that worked the best in qPCR, you saw that translated to dPCR. And now, as we go further, I think about how this could be used in plant pathology, and I think in the general sense, qPCR is our starting point because, if we're just tracking general quick testing, for pathogens, but I think when we get into, fungicide resistances, single allele changes, I think that's where dPCR is really gonna come in handy with plant pathology, and I think it has a big future there. And it's interesting to see, I think diagnostic clinics, like plant diagnostic clinics are integrating it. It's being picked up, it's just gonna take some time because I think we are used to qPCR, and so I think it'll be interesting to see how plant pathologists with they integrate not only basic research, but also very applied research into the digital PCR machine.
Yeah, it's really cool to hear too. I mean, one of the trends that we see with a lot of guests here on Absolute Genius is it's not always one or the other, right? Both have their use cases, both have their strengths, and like you said, qPCR is great as a kind of starting point and looking at kind of some of the basic quantifications. But as you need that deeper layer, that's where digital PCR can really come into play. Do you see that as well? It's not necessarily one or the other, or even this over, let's say, sequencing, right? Like there's multiple tools here that can be used just based on the question you're trying to answer.
Yeah, that's a great point. With sequencing, the time needed to get those results back, especially when we're working with growers, we can run X amount of sample, I mean, a 96 well plate in qPCR really quickly relative to sequencing. And so if we're looking for fungicide resistance, we'd love to say we can get those results back to you within a few days. But even then, compared to sequencing, you are going to be a lot faster. And interpreting that result, you see one line come up, okay, that's your like it's positive for this resistance. You can kind of interpret that quickly. And looking at the literature for dPCR back when I was working on this paper, it seemed like there's a lot more viral papers for dPCR. Viral load was a big one, and bacteria. So I don't know, other than, we think about titer with that, so maybe that's part of why that's really focused on, and also human pathogens are bacterial and viral more commonly, I guess, in terms of the big ones I would think about. But the fungal ones are still growing, but a lot of times, or sorry, like there's more and more for GPCR, but a lot of times it's looking more specifically at, different clades or these different very closely related species, or again, fungicide resistance. And so I think it's going to continue developing, or maybe it will be a mix, or it will focus. Like we suggested, you should maybe use qPCR first for what this is when you're doing general detection of the pathogen, not necessarily a clade, just pathogen detection. If you want really sensitive, you could also integrate that to dPCR, but for what we need, qPCR is enough for that. When you get down to this kind of thing where you're differentiating between low concentrations of mixed samples of different species or clades, that's where you kind of want to start bringing in the big guns with dPCR. And I think that's something that people will continue to do. And even maybe things that are really like pathogens where they like knowing it's there just even a little bit, like with that kind of stuff, that's where they'll bring that in.
What do you think are next steps for this research, but also in the field, right? Is there any trends that are coming down, or anything that's like, hey, if I had 5 more years, like what are kind of the new things, or the things that are coming down in the future?
So I think with this system specifically, so if I again all my chapters are kind of bouncing all over, but if I think about this one specifically, honestly, it was nice that my pre-doc that I was awarded lets me kind of go to that one of those next steps that we naturally started to think about and comparing. So I use this assay actually a lot when prepping for that, because I wanted to make sure I had the clades the clays that I wanted to test. I had those and I kept sampling. It's nice to just be able to run that quickly and say, oh, this, and this. And I think also one with this system, again, with the P. viticula, is doing like population genetics to see these these species worldwide, or even just in the United States. We haven't done a large population genetic study looking at where the clades are very like well sampled either. So I think it'd be interesting to kind of go further into that and look into why are they different? How are they different? Even within the same clade or cryptic species, how genetically diverse are they within locations? We don't have downy mildew out in California really at all. And so, that one kind of gets missed. But that would be really interesting if it did spread over there, how different it would have to be genetically to survive out there. So I think there's a lot of interesting work that can be done on this system specifically, especially knowing that it's an obligate. And obligates are a fun time genetically. There's just with host specificity and such. There's so much more to go in there. It just people often have resisted it because of the obligate nature. It's a pain, right? It's a pain, so people avoid it.
Taking a quick break from our conversation to tell you about the Applied Biosystems QuantStudio Absolute Q dPCR system. This instrument enables absolute quantification of your targets without the need for standard curves in about 90 minutes. Digital PCR can be as simple as preparing your samples, loading onto the plate, and running the instrument.
That's right. The system enables consistent digitization of more than 20,000 microchambers with under 5% dead volume and low percent CV. And Thermo Fisher Scientific has a suite of dPCR assays for applications like liquid biopsy research and wastewater surveillance. You can learn more at thermofisher.com/absoluteq or visit the Absolute Genius webpage. Again, that's thermofisher.com/absoluteq or visit the Absolute Genius webpage. The Applied Biosystems QuantStudio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures.
And now back to our guest. Okay, back on track here, Career Corner. Yeah, we just have to talk a little bit more about your journey and how you ended up, in this field. And I know you touched on it a little bit, but I'd love to get a little bit more in depth. And something that I wrote down from the very beginning was you said that you've always loved plants. And I was just curious, What is it about plants? Because I know, when we're all growing up, especially, me, I was really into animals, and I think that's a pretty common thing. So, what was it about plants that kind of pulled you in that direction, even when you were younger?
I think when I was young, honestly, I wrote my college entrance essay about this. It was talking about how I think at that point, really, that was a really simple mindset. And it was, I can come home from school, have a hard day at school, whatever. You go out to my garden that I had that I grown from seed or whatever, and you see like some of the plants are kind of sad, right? Or it's messy out there because you hadn't been out in a few days, whatever. But you could go water the wilted tomatoes and they'd bounce right back within 30 minutes. And it's just like I think at that point in my childhood, it was kind of you have some control over something that is you don't fully understand it. And it was also something I enjoyed that was pretty and you could produce something from it, and it brought not only me joy, but it brought, like I remember giving friends and family members sunflowers that I had like, grown, and it was just, like, getting to try and, like, connect on that, and then you meet so many different people through that too, and, just I don't know, I think it just fascinated me. I think that's just kind of a cool world that is so at that point to me was so untouched, and it also brought me joy, like, foundationally, it brought me joy, and I would hang out there with my dog, and he would lay in the sun, I would tidy everything up, and at the end of the day, you're like, "Okay, I feel so much better now." It's kinda cool to see how the full circle came about.
No, that's awesome. I mean, I think not a lot of people get the opportunity to delve into what they're passionate about at such a scale and make it their life, right? So I think that's really cool that you're able to do that. Throughout your educational journey and your career, have there been mentors that kind of helped you really cultivate, pun intended, your love for are there any moments that you kind of look back on and you say, "Oh yeah, that was really like a moment that kind of set me on this path."
So, I focused on, like, my PhD, I wanted, like, diverse experiences. I did a internship with 4-H, but it wasn't necessarily related to plant science at all. I just wanted to get into 4-H to learn. And I had a mentor there who went to my or lived like from my hometown. And so she took a chance on me to throw me into this world of extension in 4-H. I learned so much about independence and the grit of people in agriculture. And I respected it a lot. And I think in some way that fueled me into where I am now in terms of the respect I have for scientists and for farmers and growers and all these people that I think that sometimes the common public doesn't realize exists. And so there was a combination there where this person took a chance on me and let me see this world because she realized that if I could see it, then I could hopefully do something with that. And, I knew I wanted to be a scientist in terms and not necessarily work with 4-H, but I think they all kind of connect. And now I have a respect for that where I didn't have before. I also was an ambassador in my college at Ohio State for the ag college. And I think that experience also, while these are not necessarily directly related to research, I think they taught me how to be curious. And that is so important in science. Like, once your curiosity dies, like, what are you doing? Right. And it does happen, right? We get burnt out. And I think having really cool support systems of people, again, in very diverse fields of ag or, forestry or just so many things is just helped me get to where I am and have a really diverse perspective that I definitely lacked when I was younger.
You've touched a little bit already on diversity and kind of keeping, I mean, I guess it would depend on what you're going into, but this idea of like leaving yourself open for diversity and what you're looking at. And then also this structure and surrounding yourself with people who support what you're doing. So, I mean, is there anything else specific that you would just say to someone who maybe wants to go into the field that you're in?
It's definitely like having support, right? That is for sure. And, going into this specific field, I guess, is a harder question because honestly, I think it would generalize across any field, especially in maybe in plant sciences or agriculture is, yes, be open to trying different things, but also try to realize why you're doing it. I think sometimes, especially at this point where things are hard, I'm trying to write, ideas aren't flowing, or I'm frustrated, right? It's okay. Why are we doing all this? Can we get to the next step? And I think a lot of times, on a very practical level, it's important for people wanting to get into higher level research. It's important to think, okay, are you going to get where you need to get by doing these degrees or by doing this. Like, make sure you remember, even just like once you get into the actual projects, you need to remember what the point of them is. You need like to determine what tools you're going to use, what the end goal is, and you need to remember it. I think like we forget why we're doing things, and then we just get so bogged down. And I think it's sometimes really helpful just to pull yourself back out and say, okay, where am I going from here? And am I on the right path? If not, do we need to change again and redirect and think about it in that way?
Yep, that makes perfect sense. It's easy to get down into the weeds.
I use that line too often, but that's another good pun. Morgan. Oh my gosh, Morgan's mom, my husband's mom, loves puns. She's gonna love this. If you keep one of these in here, she's gonna love your puns.
The inspiration is just flowing today. Um, yeah, I just have one more question, and it's kind of a random question, but I was curious. Do you have a favorite pathogen?
I mean, I kind of love downy mildew. Like, it's a part of who I am now. Like, I honestly think I have more of a favorite plant, but also I don't because I have like a list of 12 different favorite plants. But for my wedding bouquet, it was a hosta. Like, we made hostas go on my wedding bouquet. So, favorite pathogen is probably just going to be the one I work on, but because I feel like I have to. So, I don't know that I actually have one in terms of like, ooh, this was really cool to learn about. There's some cool ones that like infect that are just fungal pathogens that infect like ants. So it's like the one from The Last of Us where like it controls their mind. And so like that's where they took it, right? But I don't know if I actually have a favorite, but maybe that makes me kind of lame, but I think it would be the one I study.
You talked about how gardening kind of got you into the whole world of plants and how it was an escape for you and just a really nice way for you to spend your time and connect with nature. And I wonder now that you spend all of your time just really deep into the plant world, does gardening still hold the same escape for you? Or are you starting to feel a little bit less connected to it than you were just because of how much time you spend on plants?
I honestly don't get to garden right now because I lived in an apartment and we did a little bit, but it's not the same. Grad school, I lived in an apartment, had a small balcony, and then we just moved to a rental house. And my husband also loves growing everything. So not if I start to lose it, Morgan would bring it back. I do want to have a big garden. I talk about all the cool gardens I want to have. I want to have a hosta garden. I want to have a hydrangea garden. I want to have a big garden of all these different tomatoes, right? So I think it's still there. I need to find it in terms of actually get to do it again. Um, so yeah, I haven't lost it. It is a lot more driven by science now than when I was 13. Um, and also sometimes with the pathology side, sometimes you'll get or even insects. Like we were some kale this semester or this year, and we came back from a trip and it was covered in bugs, right? And I was like, no, we're just getting rid of this. I know what this takes to fix this problem, and it's the end of September. I'm not fixing this. No. So I think you start to have like those trade-offs more now but the fruits of your labor are actually still as exciting.
Well, I feel like on some level too, it gives you a greater appreciation in like a new way because you understand so deeply how it works now and everything that has to happen in order for these plants to grow and thrive.
I'm horrible at any plant stuff. I would love to do it, but I tried to grow a green tea tree and I didn't start because it takes like 5 years or something from a seed, right? So I started from like a 3-year-old plant that I bought. And they're not very big because it takes a long time for them to grow. I mean, I could sit it on my windowsill. It wasn't very big at all. And I got like 6 months in. I was so excited. I was seeing new leaves come off, and I was like, "All right, couple years, I'm going to be able to, take the leaf buds and make my own tea." And then I accidentally left it outside in the summer, and it was on the hottest day of the year. It was 116 Fahrenheit. And I just forgot. I got ready for work. I left for work, and that was it. And I came back, and it looked like I set it on fire. Like literally, it was black and charred.
That's amazing though. I think it's so relatable. I kill things all the time. Like I want to propagate cool things and then I forget about them because I've got 90 other things to do. And so that's why I like growing things outside too. Like it's different when you have rain and just soil. So much better than growing things and you have to touch them every single day.
Yeah, and here's me, never grown anything, a day in my life, and I'm
Yeah,
like, I'm
take
gonna take something that's so
some
sensitive
tea.
and like the tea.
That was a bold choice.
I can do it. It's fine. We
Get
live
a
in California. You
cactus.
can grow everything here. I've killed cactuses too. So,
I also have killed cactuses. Yeah,
yeah.
it's not. I grew a cactus from seed in my AP Bio my junior year, and that thing still alive. It's
Oh,
like
nice.
it's
That's
my one
awesome.
testament as a horticulturist.
Well, Lexi, thank you so much for joining us on today's episode of Absolute Genius. We are thrilled to talk to you. We're thrilled with your stories. You were an unbelievable guest. We
Ooh,
enjoyed every minute.
Adam.
You can push me up a cliff if you want. That's totally fine. But thank you so much. Really appreciate your time.
Thank you so much.
That was Lexi Heger, PhD candidate in Michigan State University's Department of Plant, Soil, and Microbial Sciences. Thanks for joining us today for the first in an amazing slate of geniuses that we have lined up for you this season. Make sure to subscribe so you don't miss a single conversation. Stay curious, and we'll see you next time. This episode of Absolute Genius was produced by Sarah Briganti, Matt Ferris, and Matthew Stock.
Oh, don't make me say plant punster one more time. This is a tongue twister. Good lord.
I'm rooting for you.